Today our topic is nucleotide. What are nucleotide? Let's see. Whole organism, system, organs, tissue, and cell. Now cell have DNA. DNA is made of nucleotides. Today our topic is nucleotides. Now we can see what nucleotide is made of. Nucleotide is made of nucleosides. What are nucleosides? Nucleosides are again made of number one, 4 types of nitrogen bases for DNA. 4 for DNA and extra one is for RNA. Number 2, 5 carbon sugar. 5 carbon sugar. Okay. 4 types of nitrogen base, 5 carbon sugar become together, and this is nucleoside. Now nucleosides plus phosphate group, so it's become nucleotide. So there is nitrogenous bases with 5 carbon sugar is nucleoside, plus phosphate group, it's become nucleotide. Yeah, so number one is nitrogen bases. How many nitrogen bases we have? In total we have 5 nitrogen bases. For example, one is adenine A, 5 carbon, 5 hydrogen, 5 nitrogen. Number 2, thiamine T, that is 5 carbon, 6 hydrogen, 2 nitrogen and 2 oxygen. Number 3 is cytosine C, we have carbon 4, hydrogen 5, nitrogen 2 and 1 oxygen. Number 4 is guanine means G, have 5 carbon, 5 hydrogen, 5 nitrogen and 1 oxygen. Number 5 is uracil, mean U, carbon 4, hydrogen 4, nitrogen 2, oxygen 2. Now here we can say A always have pairing with T, C always have with G, and A with U in RNA. U is in RNA. All of these nitrogenous bases are divided into two groups. Number one is purine A and G. Number 2 is pyrimidine, that is CUT. CUT means cut one ring. Yeah, CUT, cut one ring. Nucleosides. Nucleoside with sugar. This is nitrogen base plus with sugar, nitrogen base, sugar, nitrogen base, sugar, with sugar and sugar. So this is double ring, single ring, double ring, single ring, and single ring. The first one is adenine. What is this? Adenine. And this one is cytosine. This one is guanine. And this one is thiamine. This one is uracil. If we together with this sugar molecule, then its name will be changed to adenosine. This is adenosine. Yeah, this one will be cytidine, and this one will be guanosine, and this one will be thymidine, and uridine. This one is uridine. Adenine will be converted into adenosine because of this pentose sugar. Cytosine will be converted to cytidine molecule because of the sugar, and guanine will be converted into guanosine because of the sugar, and thymine will convert into thymidine. Uracil will be converted into uridine. Now in this side we already discussed about nucleosides. Let's see these sides. Now these sides are nucleotides. Actually our topic is nucleotides, and these are the things that we making DNA from. So this one is adenine. This one is like we can here cytosine, here we can guanine, and here is thymine, and here is uracil. So this uracil once we already talked about if we together with pentose sugar, so it will be adenosine, cytidine, guanosine, thymidine, or uridine. But there is one another things like this one, phosphate group, yeah, phosphorus group, 4 oxygen and 1 phosphorus. It will be on 5th carbon. You see here carbon number 1, 2, 3, 4, and 5. This one will be attached with the carbon number 5. This is phosphate group. And if this one is one phosphate group, this molecule will be monophosphate molecule. If there is like 2 phosphate, then it will be diphosphate, and 3 phosphate will be triphosphate. So let's see this one, adenine. Adenine is already converted here adenosine because of the sugar. But if we put one phosphate group, it will be converted into adenosine monophosphate. What will be the name? Adenosine monophosphate. Adenosine monophosphate because of one phosphate sugar phosphate here, and this one will be cytidine will be converted into cytidine monophosphate. Yeah, this black color can see easily. Yeah? This one I think is a little difficult to see. So next one will be guanosine monophosphate, guanosine monophosphate, and third one will be thymidine monophosphate. And the next one will be uridine. Why we call it uridine? Because of also sugar. Yeah? So uridine monophosphate. Once we make this nucleotide with a single phosphate group, this is the actually nucleotide, but this is not enough for make DNA because every new nucleotides need three phosphate group. If this phosphate have 3 phosphate group, 1, 2, and 3. If there is 3 phosphate group, then it can be attached with the before one, and this one also as well as also everyone. This is phosphate 1, phosphorus group 1, phosphorus group 2, and we should be add with everyone 3 phosphorus group. Yeah? Oxygen 4, P1, oxygen 4. So this will be nucleotide for make DNA. So with 3 phosphate will be this will be like a triphosphate, adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, AMP, ADP, or ATP. ATP is kind of energy. Yeah. So this is everyone will be like the same. This one will be CTP, cytosine triphosphate, or CDP, cytidine diphosphate, and this one will be CMP. This one is the same also, like this one will be G, guanosine triphosphate, guanosine diphosphate, guanosine monophosphate, and this one is thymidine triphosphate, or thymidine diphosphate, thymidine monophosphate. This one will be uracil triphosphate, uridine diphosphate, or uridine monophosphate. So look at here, this all is energy rich compounds like ATP, CTP, GTP, TTP, or UTP. Once we have all of this ATP, CTP, GTP, TTP, and UTP, then it's very easy to make a DNA. Let's see here, number 1, 2, 3, and 4. These 4 are used for DNA. This one is DNA, and the 5th one will be convert with thymine. This one will be convert with thymine in RNA. If we are making RNA, uridine monophosphate or uridine triphosphate will be take place of thymidine. Yeah. So that's all, and now we talk about the bonds between these molecules. Quick review: DNA nucleic acid structure and organization. We are talking about DNA structure, which are made of nucleoside and nucleotide. Nucleotide is made of nucleoside, and nucleoside is made of nitrogen base plus sugar. If we have 4 nitrogen bases, these 4 nitrogen bases together with pentose sugar, that's called nucleosides. But if we are put phosphorus group and the 5th carbon place, that will be nucleotide. And if we put a lot of nucleotide together, that will be single standard DNA with triphosphate nucleotides. So let's see here, phosphate group backbone. Phosphate group are phosphorus 1 and oxygen 4. Pentose sugar is the middle between phosphorus and nitrogen bases. Deoxyribose will be DNA and ribose will be RNA. This is a sugar name. Like deoxyribose sugar will be always used for DNA and ribose sugar will be always used for RNA. And the position carbon number 2, if there is oxygen, that will be ribose sugar, and if there is miss of oxygen, that will be deoxyribose. So look at here, adenine, thymine, cytosine, guanine, uracil. It will be converted with the name of adenosine, thymidine, cytidine, guanosine, or uridine, if once we are put this pentose sugar with this nitrogen bases. Once we are also pentose sugar plus also phosphorus group, this will be adenosine monophosphate. If phosphate are one, phosphate group are only one, that will be adenosine monophosphate, thymidine monophosphate, cytosine monophosphate, guanosine monophosphate, or uridine monophosphate. But we need phosphate, almost 3 phosphate for making DNA. So A will be always pair, A and G. A will be always pair with T, A, T, and GC in RNA, in DNA. AU and GC will be in RNA. U will be take place of T in RNA. So AG are purine, double ring. Purine means double ring. And CUT are pyrimidine, single ring. Let's talk about the bonds. Phosphodiester bond covalent phosphorus between sugar and phosphorus group. Hydrogen bond always between bases in double helix DNA. AT hydrogen have 2 hydrogen bond. AT hydrogen 2 bond replication region. It's also called replication region. Which double helix DNA are reach of AT area, that area are called replication region. And which CG, which area of DNA are reach of CG, that there is 3 hydrogen bonds. So that's a strong area. 3 hydrogen bond are more strong than 2 hydrogen bond.